Right from my childhood, I was very fascinated to watch the channels like National Geographic and Discovery Channel, in which the researchers used to travel in cruises for collecting samples. I used to be very excited to see those videos, and then gradually as I became adult, I found that research was my passion too, and that's how I got to become a marine scientific researcher. In my view, I have two answers for this particular question. As an individual, I feel like I have overcome certain limitations which I had in my mind of traveling alone, coming so far away from my home, and chasing my dream. So that was our biggest impact when I consider my personal point of view. But as far as the scientific point of view, I learned a lot of new skills which I could impart to the next generation, and that's what is creating an impact from this postdoc. I expect to see more of the scientists utilizing the skills and exploring new technologies, and which will help in developing taxonomy as a science. Using basically two kind of technology. One is developing the software for creating a 3D database, image database of the animal, of the protozoans I'm identifying. And apart from that, I'm also using the technology of stereo zoom microscope, which will help to produce good quality images, which will highlight the significance of the structural differences in the organisms. The research I'm involved is based on the biodiversity assessment of foraminifera, a very interesting group of organisms, which are they actually contribute to more than 50% of the meiofauna and macrofaunal abundance and biomass in the deep sea. So a lot of them are still hidden in the abyss, and we need so many of us, so many of the taxonomists, to discover them and identify and describe them. So I think that this comes directly under the MSR action plan in supporting the research strategy priorities 1, 2 and 6, to build a strong database on the deep sea ecosystem, on the information on deep sea ecosystems, and also to standardize more innovative technologies, and also to strengthen the next generation capacity building aspect. I have tried to identify a total of 25 sediment samples so far, and from that more than 98 species have been identified and almost 85 images have been acquired. With this, I am trying to build more of my work and develop a software in which we can have an image library of deep sea species. My dream is to learn more about foraminifera and to apply them in pollution monitoring studies. And another one is that I want to make a culture lab in our country, in my country, India, and then to train the next generation of students in foraminifera taxonomy. That's my dream. For biodiversity and humankind, I want to describe and identify more and more species which would be there in the inventory, which could be used by the future generation of the students, and also to train them and apply the knowledge of the forams in pollution monitoring studies. I want to do that.